The last time we were together, we finished up by looking at this slide, which shows us an image of the enzyme that performs transcription in bacteria called RNA polymerase. Remember, in this image, we can see the double helix of DNA shown in red, and then we can see this huge protein molecule that represents the enzyme. Remember that RNA polymerase in bacteria has two parts to it. It has a regulatory subunit called sigma, and it has a catalytic subunit, or the part that actually does the work, and that's called the core. So the way this occurs in bacteria is that the sigma subunit, the regulatory subunit, is what actually guides the core to the DNA molecule. And remember, the DNA molecule isn't going to open up everywhere. It's only going to be opened up where the genes are that the cell needs to transcribe at this moment in order to eventually produce protein that the cell has a need for. So the sigma is going to bring the core unit to what's called a promoter region. This is a region or a sequence in the DNA that the sigma actually recognizes. This is located on the non-template strand, or the coding strand. So this is going to be on the complementary strand, not the template strand. And this promoter is going to be about 40 to 50 base pairs long. Now when we talk about DNA, 40 to 50 base pairs isn't very long. So the promoter region is a short segment of DNA. And it has two places in it, within these 40 to 50 base pairs, that the sigma is actually going to bind. The first one is called a TATAT box, T-A-T-A-A-T, and this is located about 10 bases upstream from the transcription start site. Now this is brand new language for us, I realize, but if you'll be patient, I'll show you in an upcoming image what I mean when I use terms like upstream and downstream and start site. So in addition to the TATAAT, the -10 box, there's another short sequence of bases called TTGACA, and that one's about -35 or 35 base pairs upstream from the start site. Now, there's obviously some additional bases in the promoter. We've got 6 of them accounted for at -10. We've got 6 of them accounted for at -35. That leaves a lot more bases. Well, those additional bases inside the promoter and outside the boxes vary tremendously. So here we are back with an image that shows a molecule of DNA interacting with RNA polymerase, both the sigma subunit shown in blue and the core shown in green. Now the additional material we have on this slide is down at the bottom. Here they're showing us that molecule of DNA again. But what they've done is they've taken a knife, if you will, if you can imagine it, and they've cut this RNA polymerase enzyme in half. So we're looking at the inside of the RNA polymerase enzyme right here. You can see that the sigma part of it is still intact. We haven't cut into that one, but we've cut into the core region. And we've done that so that you can see these darker green areas in here, these channels or tunnels within the inside of the core of the enzyme. You can also see these light green areas that are jutting out into the tunnels. Those will become important to us in a minute. But I want you to take a look at the DNA as well. The red part of the DNA remains in a double helix, but the blue part and the purple part particularly are important to us because this is the region that sigma recognizes, that sigma, this regulatory molecule, recognizes and binds with. Particularly these purple areas. This is the -10 box, and this is the -35 box. And everything in blue is the promoter. So look just above this region, and you'll see the double strand of DNA, and then this bottom strand represents our template strand, And the top strand represents the coding strand or non-template strand. It's in this coding strand that you'll see these two regions, TA, TAAT at the -10 position, and TTG, ACA at the -35 position. So again, the place where these minus terms come in is due to the fact that there is a +1 site, and that's the site where the promoter sort of juts into or bumps into the red part of the DNA. This is the part that's actually going to be transcribed. Okay, so the promoter is almost like a docking station. The promoter is the place where sigma is going to bring the core in so that it's placed in a position where it can then move forward and transcribe the important DNA at that moment. The DNA that contains the genes that code for protein that the cell has an immediate need for. So sigma comes in and binds to these regions of the promoter and guides the core in so that it too can come in and be in place and prepared to start transcribing the downstream DNA, the DNA off to the right of the promoter. Okay, so we have sigma in position, bound to the promoter in the bacterial genome. We've got the core guided in, and it's in position now to begin this process of transcription. So in our 3 steps of transcription, initiation, elongation, and termination, we are right into initiation. Sigma binds to the promoter region, and then our helix is going to open up. The template strand, the strand that's going to be used to produce the mRNA, is going to be threaded into one of those channels in the core part of RNA polymerase. Into another channel will start to stream our active monomers, our NTPs, nucleoside triphosphates. And they're going to come up into a channel that guides them towards the active site. That's the place where the action's going to happen, the catalytic activity happens, where each of these incoming bases will be matched up in a complementary fashion to the template strand DNA, and covalent bonds will be formed. Now, it's important to notice that after sigma binds to the promoter and gets the core where it needs to be, so sigma will bring the core by binding to it up to the region on the DNA where it needs to be, the sigma part of the enzyme is actually going to be released. It makes sense because it's done its job. It's the regulatory subunit. All it was needed for was to get the core where it needed to go. So, initiation is how transcription begins. And in this slide, once again, we can see a molecule of DNA. We can see the blue molecule called sigma, which is a portion or a subunit of RNA polymerase. And we can see the green molecule, which is the core. The sigma and the core have been cut in half so that we can see these tunnels or channels inside the core. The area in blue on the DNA represents the promoter, and the areas in purple represent the -10 and the -35 box, sort of the docking stations where sigma comes in and binds. So, initiation is the process where sigma binds to the promoter, gets the core in place to begin this process of transcription, and then is released. The other thing that's going to happen during initiation is that the double helix is going to open. Sigma allows for this positioning of the template strand into one of these channels within the core, and it's through that positioning, through the channel, that the DNA will be broken open. Remember, where you see blue here, this represents promoter. This is not a part of the DNA that's going to be transcribed. That begins at this +1 site, which is where the blue region meets the red region. So it's here that we need to break open the double helix, here at the active site, where we're going to start taking these incoming NTPs, and the polymerase enzyme will add them on in a complementary fashion to the template strand of DNA. A couple of things to note. Most bacteria actually have several different types of sigma proteins. And that makes sense because each one will have a slightly different structure and therefore a slightly different function, and it will allow for slightly different promoter sequences in different regions of DNA. That allows for different genes to be recognized and transcribed at different points in time. By controlling which sigma proteins are produced, the cell can help control which genes get expressed. So now we know about the specifics of initiation in bacteria. We know that sigma comes in and binds to the promoter and guides the core in so that it's in place to start transcription at the +1 site. We also know that sigma helps direct the template strand into the channels in the core and allows for the DNA helix to open up at that +1 site. Then the sigma gets released because it's no longer needed. It's already performed its regulatory work. Now we're ready to move into the second part of transcription, which is called elongation. During elongation, the RNA polymerase is going to move along the DNA template and produce mRNA in that 5' to 3' direction. In other words, the incoming RNA monomers are going to be added on to that 3' end where the hydroxyl group is exposed, and the enzyme can help build a covalent bond. Now, I showed you a minute ago that inside the channels in the core, there are a couple of regions of protein that actually jut out into the channel region. These structures are called the zipper and the rudder. The zipper is a projection of the protein that helps open the helix. And the rudder, just like a rudder on a boat, helps steer the template strand away from the non-template or coding strand so that the two stay apart from one another. So again, if we look at this image, we have finished initiation. So all we see left in terms of our polymerase enzyme is the core. The blue sigma part of the molecule is gone. We can see our upstream DNA. This is where all the promoter information is. We can see the +1 site being where the helix is opened up. And then down here we actually have our downstream DNA, which is the DNA that needs to be transcribed. Where the helix is open, we can see our zipper and our rudder projecting into this channel or tunnel within the enzyme. So these are just physical structures that physically project into the tunnel and allow for the helix to be separated and kept apart. Down in the bottom, we can see these little yellow subunits coming in. This represents the activated NTP molecules, and they're going to be fit in a complementary fashion onto the DNA template strand. Now, there is another channel up here, which you can't see in this image. You can just see the ghost of it in that the RNA molecule is traveling through it. This is the growing RNA that's being produced during this process of elongation. So, while the zipper and the rudder work, NTPs will be added to the growing chain at a speed of about 50 per second. So I want you to think about that for a minute. This enzyme is capable of catalyzing the formation of a phosphodiester bond 50 times per second. That's a pretty amazing rate if you think about it. That's something that enzymes do for us. They allow reactions to occur in real time, non-spontaneous reactions to occur in real time. And remember, they do that by doing two things for us. They bring the substrates together, They open up the DNA helix and bring those NTPs right in contact with it. And they also lower the activation energy. They lower the activation energy, which means they lower the energy that's required in order for these bonds to be built. So, during this second process of transcription, the elongation process, all of the channels within the core are full. They're busy doing something. This again tells us that the structure of a protein is directly related to its function. Those channels within that core have a job. One of them holds the double-stranded DNA, and the rudder and the zipper help keep the strands separate. One channel holds the incoming NTPs, and a third channel holds the growing mRNA strand. Remember, structure of a protein directly relates to its function. When the RNA polymerase gets to the end of the DNA template strand, it's time for the third phase of transcription, which is called termination. The RNA polymerase is eventually going to hit a stretch of DNA that functions as a termination signal. And this is the way it works. The RNA polymerase is going to get to a stretch of bases that code for a length of RNA that will have the sequence of bases that allow it to fold back on itself. Let me say that again. The RNA polymerase is going to get to this termination signal, this region on the DNA strand that has a specific order of bases that allow it to fold back on itself. The RNA that comes off of that termination signal is going to fold up on itself and create a hairpin loop. That hairpin loop is not going to fit nicely in the channel where the rest of the RNA is. It's going to disrupt the core, and it's going to stop this process of transcription. And that's what you're seeing in this image. You can see the core again cut in half. You can see the darker green channels. You can see the incoming NTPs. You can see the existing molecule of DNA that's being transcribed, the rudder and the zipper. You can see the growing strand of RNA. Most of it is out here. And here's our termination signal. This stretch of RNA at the end of the gene that needs to be transcribed is actually going to fold over on itself and create a hairpin loop. And this is too large to fit nicely within the channel in the core, and it will disrupt this process completely and end the transcription event. The DNA molecule will be released from the rudder and the zipper. That will allow the helix to come together again as a helix. The RNA molecule will be released, and it will be allowed to go on now and be further translated, and the process of transcription ends. This is a good place for us to stop. Our next video will actually describe the last bit that we want to cover as we discuss transcription, and that's how eukaryotic transcription differs from transcription in bacteria.